Now it is.
Now it is?
Yeah.
Yeah, it is.
So
Okay.
good morning to our YouTube community. My apologies. I talked for a couple of minutes before opening up the YouTube streaming. So welcome you all as well. We were just saying that we have Liam Dougherty talking next week. And today we have with us Lauren O'Connell. Lauren is, as of now, her institution hasn't updated it yet, but she's an associate professor at Stanford University. She's not an assistant professor any longer. She's just got tenure. So congratulations to Lauren for that. And she did her first degree in Cornell University. Then she went on to do his PhD at Texas Austin with Hoffman. And that's when she started working on frogs and lizards. And followed by what I thought was a very interesting, I think it was a 5-year stay as a postdoc at Harvard. So my suggestion to my junior colleagues and early career scholars is that my experience is that the longer you can stay a postdoc, please do, if you want to develop a research career before you start committing to services and teaching. If you can afford that, stay a postdoc. That would be my suggestion. Lauren spent 5 years, studied her lab at Harvard, and then moved to Stanford, where she was, I think it was 2016 or 17. She's been there ever since and doing a huge range of things. To mention the somewhat unusual, I was telling her that I really liked her website. I strongly suggest you check it out. Of course, it has the usual components, but also some unusual ones, like her sharing with us the kinds of tools that the lab has described. She has been very successful with funding. I think she has 5 or 6 current grants from NSF, NIH, and all the usual funding agencies, and a long history of funding. Of course, she's been very successful with her publications, both in peer-reviewed books, articles, and also book chapters. And again, some interesting elements in her CV. I suggest you check it out. I really liked the way you described, Lauren, all the training she's done for becoming a better mentor. So that was very interesting in my own view. Today, she will be talking about a biparental care in a frog. I want to remind you guys that we've already had at least 2 colleagues who talked about amphibians. Eva Fisher in May 2022 presented on mechanisms of behavioral evolution, lessons from poison frogs. Eva was a postdoctoral fellow at the time in Lauren's lab, and she kind of shared with us a comparative perspective on this system. I think that Lauren is going to do something different, but I just wanted to mention that you can go and listen to Eva's talk to add, to complement to what Lauren will be discussing with us today. So with that, I think that, Lauren, I would suggest that you take it from here. Thank you so much for joining us. We all look forward to what I'm sure it's going to be a fantastic presentation.
All right. Thank you. Let me get all set up. All right. Does that look good?
It looks, I mean, we have video, and I don't know if we should have sound yet.
Okay. There's no sound yet.
Okay.
Yeah.
So all yours.
Okay, perfect. Well, thanks for letting me or inviting me to come give a talk. I think this is a really great community and a great seminar series. And I've really enjoyed watching all the things on YouTube. And so yeah, I'm excited to share some kind of updates from our lab and what we've been working on in the last couple of years. And so what I want to focus on today is our investigations of kind of family relationships. And so in this picture, this kind of title slide picture, what you see is this couple. The male is here, the female is here, and this kind of blurry blob in the background is their tadpole that's begging for food. And so I'm really interested in the relationships that are taking place here, which parallel a lot of like the family relationships that we see in a lot of taxa. So these parents have to work together to coordinate feeding. The offspring, the tadpole, has to communicate their needs to their parents, and they all have to work together as a cohesive family unit. And so I'm really interested in how these dynamics play out and understanding these bi-directional relationships. And so the main system I'm going to be talking about, I'm going to really focus on one species today because we're going to go in a lot of depth in kind of mechanistic understanding of how these behaviors are reinforced. But I just wanted to give a plug for poison frogs in general, because I think a lot of people don't really, when you think about parental species, you know, amphibians are not the thing that comes to mind. And so one of the reasons that we focus on poison frogs is because I think they have, you know, a lot of variation in different kinds of parenting or reproductive strategies. And so I've given you the phylogeny here. I've highlighted paternal species in blue, and then you can see that there have been some independent transitions to species where only females take care of offspring, or species that are biparental. And so I think it gives us this kind of model clade approach, gives us a lot of power in understanding how different behaviors and the underlying mechanisms have evolved to support different reproductive strategies. And of course, you know, poison frogs are also sometimes poisonous. And so the other half of my lab doesn't do neuroscience, but they do kind of this study of the evolution of chemical defenses. And again, this model clade approach has been really powerful in understanding how different traits have evolved. And so you'll see that some poison frogs are toxic, and some of them are not toxic here, which is shown in black. And this kind of, it's a good example of how like evolution has been able to teach us some fundamental principles of how traits evolve. And so the example I just wanted to share with you, if you're interested in looking at this later, is that we've used this comparative or model clade approach to discover how poison frogs protect themselves from their own toxins by evolving these toxin sponge proteins. But I'm not going to talk about that today. I'm going to talk, we're going to focus on parenting. And I want you to notice that a lot of these parental or like where females tend to get involved in parental care in amphibians tends to be in these toxic species. And so these traits might seem very different, but I think they're actually kind of tightly related. And we think this is because some species of poison frogs, they're able to chemically defend their offspring earlier in development. And so this could be an additional reason why poison frogs that are toxic have evolved these different chemical strategies, is because this is an important role that the mom plays. And so, you know, these traits are tightly linked. And so we've used this model clade approach in the past to understand the evolution of parenting. So for example, we've looked across these two independent evolutionary origins to find, to look for brain regions that are involved in parenting. And we found a lot of the similar brain regions that are involved in promoting parental care. But sometimes we find that there are different signaling mechanisms. And so I think it's another nice thing about taking a model clade approach is that it allows you to find kind of alternative mechanistic strategies to promoting similar behaviors. And so, you know, but also we've been able to look at very conserved traits. So for example, in mice, Catherine Dulac's lab has found that galanin promotes parenting, and we have found the same in amphibians. And so there are lots of opportunities to discover new mechanisms, and there are also opportunities to look at kind of conserved traits that are common across like kind of many different parenting animals. All right, and so I want to dig a little bit more into kind of a general intro into poison frogs. Because their behavioral diversity is very much linked to their environment, and so linked to their ecology. And so we have a bunch of different species in the lab that show different parenting strategies. So for example, there are species where mostly males do parental care, and the parental care I'm showing you here is they transport their tadpoles. So there are these little tadpoles on their back. There are some species where only females do this, and then there are some that are biparental, where both mom and dad are doing the parenting. And who does this behavior is linked to resources in their environment. And so mostly what we think is going on is that it's linked to this variation in pool size. And this was first proposed by Kyle Summers, whose career really laid out the behavioral diversity of poison frogs in a really lovely and powerful way. And so, for example, this species right here, Alabates femoralis, they put there a lot of tadpoles. They transport them all at once. So they do this school bus method of moving offspring around. They put them in these really big pools. And everybody grows up, kind of gregariously together. And that's kind of the extent of parental care in that species. But as we're moving forward and these adults are using smaller and smaller pools, we get the evolution of these other kind of offspring related behaviors. So for example, the tadpoles become cannibalistic, and this is where you see species mostly transporting one tadpole at a time, because if they transport two tadpoles to a pool, there will only be one later. And so there's this kind of coevolution between the number that's transported and the evolution of aggression or cannibalism in the tadpoles. And then you come to these really, really tiny pools. And here the thought, and this is an idea that Kyle Summers and Jason Brown proposed, which is that the smaller these pools get, the less resources there are. There are less predators, but there are also less nutritional resources. And so what we see is kind of the evolution of more in parental investment and the involvement of moms in parenting. So moms come back to feed the tadpoles throughout development. All right, and so that's kind of the setup of poison frogs that I wanted to share with you. So there's a lot of beautiful behavioral diversity that we see, but for today, what I wanted to focus on is we're gonna just focus on this species right here. This is Rennetomania imitator. They are a monogamous and biparental frog, and they rear their tadpoles in these little pools here contained within plants. So these pools are not very big. It's about 10 to 15 mils of water. And they really have to coordinate as a team to take care of their offspring. And so I'm going to draw that or kind of illustrate for you kind of a reproductive bout of a couple. And because I think I would really want to highlight the complex family units that they have. I think a lot of people maybe think that amphibians don't have very complex social lives, and hopefully after this slide you will you'll agree that they have a lot going on. And so in this species, males and females are monogamous and they're biparental. They lay their eggs in the leaf litter. And usually dad takes care of them. He keeps them moist, he cleans them. And then when they hatch, the tadpoles are still aquatic, and so they need to get to water somehow. And so the parent carries the tadpole on its back to a pool of water. And because the tadpoles are cannibals, each tadpole gets its own little plant nursery in the forest. And so they're usually transporting one tadpole at a time to a different location. And dad has to remember where he put them, because within these little pools, what happens is that dad visits them almost every day, depending on the dad. And if the tadpole is hungry, they do this little begging display, and I'll show you a video of that in a moment. And if this signal is sufficient, dad hops out and he does this kind of special call to mom. And so Ava Fisher, who was a postdoc in my lab, who has her own lab now at UC Davis, has shown that this is, there are some unique signatures of this call when dad is trying to get mom to come feed their offspring. And then when mom comes, she dips into the water, the tadpole does this little begging behavior again. And then she lays these trophic unfertilized eggs for the tadpoles to eat. And then This happens, every couple days for 1 to 2 months. And so, until the tadpole kind of completes metamorphosis and walks out of water as a little froglet. And so I hope this gives you some sense that these frogs have a lot going on. Socially, they have a lot of family responsibilities, and they have to do a lot of coordination, both in communicating, parent-offspring communication, but also parent-parent communication to make sure the tadpoles get fed. And these responsibilities are really important. So for example, if you remove the male, about 80 to 90% of the tadpoles will die in some field experiments that were done by Kyle Summers. And so I want to focus on this family unit today. And I want to tell you our progress on understanding parenting teamwork. So, there's some teamwork involved in, dad visits the offspring, but it's mom who feeds. And so how do they allocate responsibilities and communicate with one another? Then we're going to look at some parent-offspring interactions, how the tadpoles recognize their parents and how they decide to do this begging behavior, which kind of gets to this idea that these relationships, these family relationships, influence tadpole behavioral development. And so the first story I'm going to tell you today is a story by Billy Goolsby. So she's a graduate student in my lab, and her thesis focuses on parental synchrony or parental teamwork. And one of the first things that Billy did when she entered my lab is that she wanted to set up, have this monitoring of families constantly, instead of these 5-minute kind of observations that we were doing haphazardly. And that proved a little bit tricky for us because a lot of camera traps that are commercially available are based on kind of body heat and are targeted to mammals and birds. And so we went through a range of different camera systems, and we ended up using these Wyze security cameras and modifying the insides of the cameras a bit to adjust the focus. And we found that they're actually a really great system for kind of monitoring ectotherm behavior remotely. So for example, the camera is triggered based on movement. It sends Billy a text message when something's happening, and she can decide whether or not she wants to go down and take a hormone sample or something like that. But we have all these cameras kind of placed above the nurseries in the terraria. And so we're able to kind of monitor all the parental interactions that happen within this kind of tadpole nursery. And so what you're seeing here is this dad is doing a good job doing these calls to mom. He's calling mom over, and he's saying hey, come feed this tadpole. It was also really important to us that these cameras worked in the field. And so we've also piloted these in the field where we've been able to capture a lot of interactions that we were totally unaware of. So for example, a lot of predator interactions with the tadpole and a lot of other interspecies interactions we're able to capture with these cameras that are then triggered by movement over the nursery. And so it's been a really nice development for us. And it's also these, cameras are very inexpensive. And so it makes them a little bit more accessible. Okay. And so after Billy kind of set up this system in the lab, one of the first things she wanted to look at was how often are parents visiting their tadpoles, and how often do they feed them? A lot of these kind of standard behavioral measurements, but not something that we hadn't really been able to do in a rigorous way before we had these camera traps set up. And so one of the first things that she found, or that she noticed, is that our lab colony is kind of divided into high care families and low care families. And what I mean by that is that these coordinated family visits where both mom and dad are visiting, there are some families that visit their tadpoles quite a lot. And there are some families that are down here that are not visiting their tadpoles very much. And Billy was able to show that there's a correlation between the family's total time visiting and the number of eggs that the mom is feeding the tadpole. We wanted to then ask, what are the contributions of mom versus dad in these coordinated family visits? And so what we found is it's really dad's behavior that's driving the differences between these families. And so what I'm showing you here is this is a high care dad. You can see that this really tracks with the visits with both of the mom and dad together. Versus a low care dad. He's not really visiting his tadpole very much. And then because of that, he's not really calling mom to the nursery site. And so what this means is that there are some pairs where the dad's not visiting very much and the tadpole's not getting as much food, whereas there's some families that the dad visits very frequently. He calls the mom over more frequently, and the tadpole gets more food because of that. And so we next wanted to ask, do moms compensate for this lack of parental investment? In a lot of species that show parental care, there is this kind of compensation when the other partner is not, is other, like, busy doing other things or gets eaten or something like that. There's this flexibility in parenting behavior in many species. And so what we found is that mom doesn't really visit the tadpoles alone. So there's no compensation for paternal investment. And so what I'm showing you here, these are mom visits alone, and versus dad visits alone. And so mom's not really visiting unless the dad is calling her over. And so it's really the behavior of dad that's driving this coordinated parental care and this investment in offspring. All right. And so just to summarize what I've told you so far, or what I've shown you, is that fathers drive this offspring investment by the number of times that they're visiting the tadpoles, and that, when they're calling over their partner. Moms don't compensate for bad fathers. So when males are not visiting their tadpoles, they don't step up to do it. And this is, something that's been shown in other species that are genetically monogamous. So there's this kind of mutually kind of assured destruction where mom's not going to compensate when dad isn't doing his job. And so we found this kind of difference or variability in parental investment really interesting. And so we wanted to follow up a little bit more and ask, what are the mechanisms, either hormonal or neural mechanisms, that underlie variability in the quality of care that's being provided for these tadpoles. And so we're going to get a little bit into some neuroscience here. And so we wanted to look at what neurons are turning on or are more active when the parents are interacting with their offspring. And so the experiment Billy did is that she took parenting frogs and she exposed them to either a tadpole that did this begging behavior and a tadpole that didn't do begging. Because what we wanted to do here is we're looking for neurons that are responding to offspring signals. And the way that we do that is we use this protocol called phosphotrap. And the idea here is when you activate a neuron, or when a neuron receives a neurotransmitter signal, it becomes more active and it needs to make more protein. And so it phosphorylates their ribosomes. And so the neuron is more active, it's making more protein, these ribosomes are more phosphorylated. And then this modification of the ribosomes is something that we can detect with an antibody. So we can label the neurons, or we can use an antibody to pull down the mRNA associated with the ribosomes. So in a way, it's like sequencing RNA specifically from neurons that are more active during a particular behavior of interest. And so when we do this process, we come up with basically a pool of genes that are enriched when they're exposed to begging tadpoles versus when the tadpole isn't begging. And so it's a way to look for genes that are kind of responsive to offspring signals. And I'm going to show you this data in a volcano plot. And things on the right are enriched in response to begging, and things on the left are genes that are depleted in response to begging. And so, neurons are making a lot of things, and so there's a lot of genes to look at here, but I'm going to highlight, just for the sake of time, a couple of things that we found that were interesting. And so what I'm going to show you this data in is this log fold change. And so I'm going to show you frogs that were exposed to begging tadpoles in green, and frogs that were exposed to tadpoles that were not begging in yellow. And so there are some genes like this NPY receptor. NPY is involved in feeding behavior. These are active in both scenarios. And so this is, this maybe just being exposed to a tadpole activates, the neurons with these receptors. But, these aren't something that we're necessarily interested in. What we're interested in are things that are enriched in parents that were exposed to this offspring signal. And what we ended up finding was enrichment of a lot of genes involved in opioid signaling. And so, endogenous opioid signaling is, the idea is that this regulates this reinforcement of behavior. And at the same time, we found this decrease in nociceptive signaling. And these are really kind of neuronal signatures that regulate pain. And so what we find is this enrichment of endogenous opioid signaling, this decrease in this, pain signaling. And so this was really exciting to us because it could be a way that these parenting behaviors become, these parenting interactions with the tadpoles become reinforced. And so we wanted to test this idea. And so this work was done by Ashlyn Callan, an undergraduate in the lab. And what Ashlyn did is she took fathers and injected them with either saline or naltrexone, which is this long-lasting general opioid antagonist. And what she found when she quantified the number of parental visits is that in low-care dads, there's not really a difference. And this is probably because they're already not visiting their tadpoles very frequently. But when we look at high care dads, then we see this decrease in parenting behavior whenever we're blocking endogenous opioid signaling in the brain. And so what this told us was that, that perhaps this could be, a mechanism where interacting with your offspring is a reinforcing behavior, or like something that, might give you motivation or pleasure to come back and do it again. And so we thought was very exciting. And I want to give you some kind of extra context to think about this data in, because there's some evidence that this happens in other species, and especially in mammals. So for example, the opioid receptor allele that some primates have kind of predicts the maternal attachment strength. So it predicts the strength of mother-infant bonds. And then also plasma opioids can also predict postpartum depression risk. And so it's starting, measuring plasma opioids is starting to be used as a biomarker during the second trimester of pregnancy to kind of predict, who might need some additional, help after delivery. And so this was really exciting to us. So there's these associations between opioid receptors and opioids in the plasma and parenting, but the neural mechanisms that underlie these interactions is not very well understood, and I think this is where we can make an interesting contribution. And so our current hypothesis is that there are differences in endogenous opioid signaling that predicts these high or low investments in parents. And so our prediction is that if you increase opioid signaling, you increase parental synchrony and the quality of care and the number of interactions you have with your tadpoles. And if you decrease opioid signaling, you have these instances of, kind of neglect where the parents aren't taking care of their offspring. Or we've seen some cases where low quality parents will eventually consume their tadpoles. And so it might increase the risk of infanticide, for example. And so these are kind of future directions and things that we're actively working on now. But I think, this phospho-trap data and then this, the pharmacology work that we did, I think points to like an interesting direction that we're following up on. Okay, so to summarize what I've shown you thus far, we've seen that parents, or that dads especially, are really important in family dynamics in this species, and that individual differences in parenting quality might be driven by endogenous opioid signaling. But kind of what I want to switch gears to now is the tadpole side of the story. And so I focused a lot on parents so far, and we wanted to see how tadpoles, how this variation in investment influences tadpole behavior and brain development. And so I'm going to switch to telling you about this work with parent-infant interactions. And this work was really driven by Julie Butler, who is a postdoctoral fellow in my lab, and she just started her lab at Augusta University to work on more of these kind of tadpole-centered questions. And so just to remind you, these tadpoles do this begging behavior to their parents. And this was also originally described by Kyle Summers' lab, where they showed they suggested that it was an honest, indicator of need, that the tadpoles were more likely to do it when they're hungry. And it was kind of an energetically costly behavior to do. And this might seem like a very niche behavior, but I actually think it reflects a very broad principle where, many infant animals have to communicate to their parents their nutritional needs, and they do this in many different ways. And so I think that, the behavior is there. It has been evolved many times, but the neural basis of this behavior and how these communication or these signals have evolved is not really known. And so I think this is where we can also make a good contribution. And so we're going to focus for the next couple of minutes on this interaction between the tadpoles and the parents, and look at kind of caregiver recognition. And so I want to show you how we think about how tadpoles decide if an individual is a caregiver. How do they recognize, moms or dads apart from other individuals, and what in the brain is happening to produce these affiliative behaviors. And so a lot of this work was driven by Julie Butler, but this was also a collaboration with Scott Edwards and Devraj Singh, and who are both ornithologists, and they really helped us think about kind of the evolution of begging signals, because there are a lot of parallels here with the tadpoles and of begging chicks. And so their contributions and kind of the concepts around these ideas was very helpful. All right. And so we first started with this question and thinking about, how does a young animal recognize their parents? Or do they just beg to anybody? And so, when we're thinking about what kind of sensory information comes to the tadpole, there are a lot of different mechanisms or, sensory systems where this could happen. We decided, and I'm only going to tell you about these two for the sake of time, to focus on vision, because there's some evidence by Corey Richards-Zawacki that suggests that tadpoles visually imprint on their parents. And so we thought that maybe vision might play an important role in parental recognition. And then we also decided to look at olfaction as well. And we wanted to see which of these caregiver-related stimuli was necessary or sufficient to elicit begging. And so what Julie did is she wanted to give them stimulate different kinds of sensory modalities, and then test for begging behavior. And so what I'm going to show you here is the number of tadpoles that display begging behavior, kind of as a percentage here. And so no stimulus or giving them like a real female, most of the tadpoles will beg. So these are like our control situations. And when we give them just olfactory information, so this is the smell of a reproductive female versus just frog water, this is also not sufficient really to elicit begging behavior. And then just the visual information alone. And so what we did here is we gave them a 3D printed or like a kind of a model 3D frog, or just this kind of gray disc for, it's not a frog, but it's, something visual to look at. It also was not sufficient to elicit begging behavior. But when things got really interesting when we started doing these kind of multimodal mixes. And so what we found was that we had both smell and the presence of the 3D model frog, this was enough to elicit begging behavior. But interestingly, this combination of the smell of mom and just a gray visual disc was also enough to elicit begging behavior. And the amount of time the tadpole spent begging wasn't different between these kind of two different conditions. And so this was really fascinating to us because it suggested that tadpoles will beg to an object if it smells like a female. So they need some like visual like goal to beg to orient to, so vision, is important. They need a goal, but it needs to smell right. And so we next wanted to test whether these sensory modalities were necessary for parental recognition. And so what we did is we exposed them to either a conspecific or a heterospecific. So these species look the same. They're actually Mullerian mimics of one another, but they have different smells, different odors. And so this is our control situation. So a lot of tadpoles will beg to a conspecific, but not a heterospecific. When the tadpoles are blinded, they also can distinguish between these two different stimuli. So this is a video of a blind tadpole. Once it found its the frog, then it started doing this begging behavior. And so it's not, vision isn't necessary to do this. But when we made the tadpoles anosmic, they couldn't tell the difference and started begging, to anybody. And this was also similar in the situations where the tadpoles were both blind and anosmic. And so it kind of reinforced this idea that olfaction is required for caregiver recognition. Okay, and so, once we got a handle on the sensory modalities, we wanted to look at the kind of what was happening in the brain to promote begging behavior as kind of an inroad into understanding how offspring might be, decide whether to or not to respond to a frog. And so we took advantage of this individual variation in how the tadpoles respond to stimulus. And because, about 70% of the tadpoles will respond with begging behavior, and the rest won't. And so what we decided to do was use this same PS6 kind of phosphorylated ribosome marker to label neurons that were more active. And then we counted the number of neurons that were more active across a bunch of different brain regions in the tadpoles. And first, I'm going to show you these sensory representations. And so what we're counting here is the number of PS6 stained cells. So you can think of this as the number of cells that are more active. And so in begging tadpoles, for example, their olfactory bulbs kind of light up. There's a lot of neuronal activity there. But this is not the case, for example, in the tectum, which regulates or processes visual information, or the torus, which processes auditory information in amphibians. And so those of you that think about mammals a lot might be kind of surprised at this, like we were, because a lot of olfactory information in adults is processed by the olfactory cortex. So it's kind of these higher brain regions that are doing a lot of decoding of different stimuli. But actually in young animals, it seems to be the olfactory bulb itself that is doing a lot of the computation or the kind of decoding of different olfactory information. And then the olfactory bulb kind of then like sets. There's this kind of critical period in rodent pups where they can, you know, recognize mom versus not mom. And this kind of like sets them as they enter puberty. And so this is something similar then that we're seeing in tadpoles, that a lot of olfactory information processing might be happening in the olfactory bulb itself. Okay, so what I've shown you so far is that we have these caregiver chemosensory cues, that they're detected by the olfactory system. But what we wanted to do is kind of draw a line between the olfactory input and the cells that are regulating begging behavior. And so we did the same thing with phosphotrap that I mentioned before with adult animals. And, you know, I'm going to show you it the same way, enriched in response to begging or depleted. And what we found, you know, we found a lot of genes that were enriched and depleted. And I really want to focus, or what we, you know, really thought was kind of a glaring or obvious result, was that there was this enrichment of dopaminergic signaling in the tadpoles that were doing begging behavior. So it was looking at, you know, dopamine is involved in reward, it's involved in locomotion, it's involved in parent-offspring interactions. And so we next wanted to see, does dopamine mediate this begging behavior output? And so we then did some, just like in the adult frogs, you know, we took this candidate from phosphotrap and then we proceeded with some pharmacology to test to see if we were on the right track. We injected tadpoles with vehicle and then looked at the amount of time begging, which I'm going to show you here on this graph. And then we injected them with a dopamine agonist or an antagonist, and we showed that the antagonist blocked begging behavior. So if you block dopaminergic signaling through the D1 family of receptors, it blocks begging. And then there's also this kind of opposing function. So I wanted to show you this off-target begging with the D1 agonist. So it's kind of, we're like kind of overactivating the system, because this tadpole is actually begging to its reflection. And so it's instead of the actual frog. But then we also looked at the D2 family of receptors, and we found that which have kind of opposing roles, D1 and D2 family receptors, and we found that the agonist here also blocked begging. And so, which suggested that like dopamine signaling is required for this behavior. And so because dopamine also regulates motor behavior generally, we wanted to look at total, you know, the total activity time, and we found that there was no differences in movement or total activity across these groups. And so what we found here was actually very similar to something that people have found in xenopus tadpoles, where signaling through D2 receptors, you know, this results in a decrease of intracellular activity. And in xenopus, this blocks swimming behavior. And we found in imitator that this blocks begging behavior, but not swimming. And then in xenopus, when you activate D1 receptors, this results in really fast swimming. And in imitator, then it results in this like really fast motor display of begging. And so there are a lot of parallels here between motor systems and dopaminergic regulation, but in xenopus, it regulates swimming, and in imitator, it seems to regulate this begging display. And so we thought that this was interesting, that there was these kind of hallmarks of motor systems involved. Okay, so the next thing we wanted to do is ask which dopaminergic neurons are important, because there's more than one set of dopaminergic neurons in the amphibian brain. So there's the preoptic area, and there's this area called the posterior tuberculum. And so we wanted to look at which neurons were actually important. And so what we did is we took a conspecific female and we took the odors of that female and then we gave them to a tadpole. And then we looked at the number of dopaminergic neurons that were more active. So we're looking at the overlap of PS6, this marker of neural activity, with TH, which is a marker of dopaminergic neurons. We also did the same thing in tadpoles that were given a heterospecific odor. And so what we found is that we're looking at the number of dopaminergic neurons that are more active. And so in the preoptic area, there are some differences. It's not quite different from control. And there are no differences in this kind of rostral population in this posterior tuberculum neurons. But things got really interesting when we looked at these caudal populations. And you can see here that these dopaminergic neurons are very sensitive to these female odors from a conspecific, and that they're not activated in this like heterospecific context. And so they seem to be very sensitive to this important social cue. And this is kind of reminiscent of work that has been done in zebrafish that looks at how these particular neurons in the posterior tuberculum are sensitive to this tactile and visual information. And this paper actually proposed that this population is like a feedback regulator of sensory systems. And so we felt like we were on the right track here because we identified dopaminergic neuron, dopamine is important, and pinpointed this one cell population that seemed to be responsive to these odors. Okay, and so this was great. Dopamine is involved, but we wanted to look at whether or not that was required for begging. And so what we did is we injected some different populations of or brain regions where these dopaminergic neurons projected to with this neurotoxin that kills dopaminergic cells. So if you inject the neurotoxin into a brain region, the toxin will travel retrograde up the neuron and kill that cell population. So it's a way to kill dopaminergic neurons that are projecting to a particular place. And we did this both in the striatum and this motor region called MLR that regulates voluntary or goal-directed behaviors. And so what we found when we did this is when we ablated dopaminergic neurons that projected to this MLR region, this totally got rid of begging behavior. And so this caudal population that is sensitive to olfactory information is required for this begging behavior. There is no significant difference in total activity time, although, this isn't statistically significant, but there is a drop here in total activity. And then we have this, and then we also looked at the striatum, and there was no difference in begging behavior here. And so what we found was that these dopaminergic projections are required for this behavior. All right, and so putting this all together, what we've been able to look at is that, we found these olfactory cues that modulate dopaminergic neurons that then project to these motor regions that are important for behavior. And so this was, if you're interested in reading more about this work, this has been posted on bioRxiv, and it's currently in review, and you can read the full paper there if you're interested in it. But what I really want to kind of end on is how parenting quality influences tadpole behavior. And so I want to bring it back to look at, whether or not this variation in parenting behavior might influence begging behavior on the part of the tadpole. And so this work was done by Penelope Baker, an undergraduate in the lab. And what she looked at is she divided these high care and low care families that I talked about in the beginning and looked at whether or not the tadpoles preferred their parents versus strangers. And so I'm going to show you this parental preference here, which is the time spent begging to their actual parents versus a conspecific that is not their biological parent. And what she found is that tadpoles from high care families prefer their parents over strangers. So they they spend more time begging to their parents versus some other individual, whereas low care tadpoles don't, you know, I somewhat don't have a preference, and even some of them prefer strangers over their actual parents when they're from low care families. And so it seems like this, you know, variation in parental care quality is then influencing the outcome or the behavior of their infants. And so I'm gonna kind of just summarize. I'm gonna try to end for the sake of time and just summarize what I've shown you so far and give you an idea of where we're going next. And so we're interested in understanding like how this variation in offspring signaling influences parenting, because we see that, you know, they'll beg sometimes to their parents or strangers. And this could be, you know, feedback onto the parents and how much they're willing to invest in different tadpoles. We're interested in understanding how this variation in parenting, like high care versus low care families, influences tadpole behavior and brain development, as well as kind of understanding this hunger state, or like how these family relationships are tuned by environmental resources, which are, I think, questions that, you know, that lots of families and different kinds of parenting and offspring relationships have to deal with. But I think that our system gives us a lot of power in looking at not only at behavior, but also at the neurons and brain regions that are actually controlling these behaviors and allow us to manipulate them in depth. And so I want to give a moment to thank my lab. I talked mostly about the work of Billy Goolsby, who's here. She's a graduate student in the lab, and she'll be looking for postdocs soon. And then I also talked about the work of Julie Butler, who started her own lab. Also would be a great person to invite to, you know, either university or onto the Fine Seminar series. She's a lovely human. And I want to thank all of you for your time.
Thank you very much, Lauren. A very exciting, very interesting presentation. I'm sure we'll have tons of questions from the audience. From those of you who may not be familiar with how we proceed in the Fine Seminar, you have the chatting window, and we ask that you simply type a question mark like Theo and Michael have already done, and I'll be calling you in the sequence that you posted your question mark. Now you go, you're pushed up if you're a student or an early career colleague. We definitely want to hear from students, so please type a question mark and an S for student, and we may go straight to you because we want to hear from the young voices in the audience. With that, once again, thank you, Lauren. And let me open the chat to see who do we have first. And I think it is Theo. Theo, if you can please, before you ask the question, if you can please let us know where you're joining us from and what's your institution.
Yeah, I'm Theo Bakker from University of Bonn. I'm Emeritus Professor of Ecology here, working on fish. I was interested in the division in low and high parenting frogs. Have you any idea how this is maintained in the population? Is there an average fitness, equal fitness of both types of parents?
Yeah, I can answer that. I'm sorry. Am I supposed to, but first I have a question. Do I need to stop sharing my slides, or?
It's up to you. You decide if you feel like you might want to go to a slide, or if you prefer to have this full screen, and do whatever you think works better for you.
Okay. Oh, somebody asked to close it. I can definitely do that. And I think I would prefer that so I can see people's faces. Okay. So the question was like, so about high-low care parents, and does this happen in nature? And so, because we have, just to be clear, we found this in the lab, and, you know, because having a lab population allows us to maintain constant surveillance. And so we found this maybe like in the last year or two. So we haven't been able to replicate this study in nature, but that is obviously kind of the next thing we want to be able to do. In the lab, the tadpoles grow up a little bit more slowly, and they're a little bit smaller when they metamorph. And so there does seem to be some effects on the tadpoles, not only for behavior, but also their development. But whether or not this is like affects then their parental investment, you know, as they're growing up, or what the fitness advantages are of these two different strategies in nature, we haven't been able to look at that yet. You know, it kind of reminds me a lot of this like high licking and grooming work in rodents, where there are these kind of two different behavioral strategies, and they have different, you know, fitness outcomes and, you know, different kind of ecological links and things like that. And so it could be similar to that, but we don't know yet. But I agree, it's important that we do that next.
Thank you.
Michael, you're next.
Yeah, hello, I'm Michael from Constance University. I'm working on parenting in birds. And yeah, I mean, first, your fantastic talk. I loved it. So I wanted to ask the same question as Theo, so I ask a different one. So basically, you showed that there's no parental compensation. So if basically the female responds to what the male does. But we see that in a lot of birds that this is happening. So could you just speculate on why we have this difference? I know this is relative, but any idea from your side?
Yeah, so yeah, because I used to look at, or when I was a graduate student, I did some studies on kind of parental compensation when one parent or the other is removed in a biparental context. And almost always there's some compensation on the part of the other partner. But, you know, in here, that wasn't the case, and we were really surprised. And so it could be the kind of function of how, or like how the relationship or teamwork works in this system. So for example, females aren't, you know, dad is the one who kind of put these tadpoles in different pools, and he's the one that's doing these checks on the tadpoles to see if they're hungry, and then he calls mom over to this site. And so, you know, mom often doesn't she can, but she often doesn't transport the tadpoles, and then she just doesn't go visit, you know. And it could be because this is a very energetically costly behavior, and she is, you know, foraging and doing other things while this is happening. And so, but you know, the other systems in which we've seen that there's no parental compensation is in these genetically monogamous species, where this is how partners kind of keep their other partner in check and kind of avoid desertion or avoid kind of cuckoldry. And so this happens like you know, sometimes in owl monkeys and things like that, is that genetically monogamous species sometimes have less compensation when their other parent isn't doing their job. Yeah, does that make sense?
Yeah, makes sense. Thanks for the answer. Thanks. Stimulating.
Maren, you said we can skip you. So, and Lauren, I know he had to go. So, Kuz, if I'm pronouncing your first name well, you're next. If you could please introduce yourself, everyone, please let us know where you're joining us from.
I'm Koos Bomsma, University of Copenhagen, and I'm very interested in social systems that have obligate monogamy, but then in social insects. But that's not what I would like to ask you about, because my question is still related, I think, in some ways to the previous two, because it puzzles me greatly why there is so much variation within the same gene pool, that you can essentially move from almost having no care to having almost the maximal amount of care. And somehow I can't escape the feeling that, even though you probably haven't investigated that part of that must be genetic, because the alternative would be you would have to show that it really is induced by breeding conditions, which I'm sure will have some effect, but I can't imagine it explains everything. So I think, simple question is, what do parents, or what does one parent get out of neglecting their young? So what are the fitness compensations that you could think of when they do that? Because the default setup from the very beginning seems to be, yes, we're in this as a monogamous pair. We're going to do all this. We spread our kids out to different sites because otherwise they will eat each other. So why neglect? I mean, is there some hypothesis of another type of compensation than you talked about?
Yeah. So I have kind of two things that I want to point out, that I've been thinking about. And if anybody else has more ideas, please, I would love to hear it. And because we've just started thinking about these things now since our undergraduate in the lab discovered these kind of relationships. And so one, I mean, it could be that dad is, so one is that in the lab, we restrict the number of tadpoles that they're caring for, so that we don't have maybe dads aren't visiting as much because they're taking care of 9 tadpoles versus 2 tadpoles. And so that we've controlled for in the lab. They only have this, 1 tadpole at a time. So, they could be foraging, in nature, something like that. They don't have very other things to do really in the lab. And so, why are they doing that? In nature, they could be out foraging or defending their territories instead of visiting their tadpoles. I think it's unclear. I think we really need to do these natural experiments in the field to be able to test that idea. And then the other thing I wanted to point out is that this is a bi-directional relationship between parents and their offspring. And what I mean by that is that, and what I didn't get to show you because I ran out of time, was that to be able to study how tadpole behavior influences the number of visits of their parents, what we've been doing is we built this tadpole robot where we can control the begging frequency. And when the tadpole when the parent comes, we can decide whether or not the robo-tad is going to beg to their parents or not. And what we found is that when the tadpole, the robot beg is at this specific frequency, this is reinforcing, and the parents will visit the tadpole more often. And so it could also be that there's something about the tadpoles that isn't their behavior that's not as reinforcing, maybe they're not begging as much or something like that. And so I think it's difficult to disentangle because it's 2 sides of this relationship. And so I think that, that having this robot will allow us to disentangle a little bit, or at least control for tadpole behavior. And so I think that's something that we weren't really thinking about whenever we were looking at these high care, low care families. So it's not just about what are the parents doing somewhere else. They could be foraging, they could be taking care of other tadpoles or something like that, defending their territories, but also the behavior of the tadpole itself could be influencing how often the parents are visiting. I guess I don't have an answer to your question. And those are the things that we've been talking about as we've been thinking about and processing these ideas.
Okay, thank you.
So I guess I'm coming next. I don't have a question, so you won't have to worry about a specific answer, Lauren. I just want you to help me consolidate the basics of this wonderful presentation on the neurological basis of parental care. So if I followed you well, as you were discussing how the endogenous opioid release, maybe the way you presented, may be driving care quality. Mm-hmm. I just wanted to have if you can please repeat to me, then what is driving the opioid release, and to what extent? I'm listening to you, and it's making me think about Shirazio's suggestions of all this comes down to some kind of homeostasis regulation. And so again, it's not a concrete question. Do you think you didn't really get into the monogamy and pair bonding between the partners? Is from what you know that these are the same systems? I mean, where both males and females are getting some kind of positive reward from providing care with an endogenous opioid release, along the same lines that it's been suggested that in some systems that may be reinforcing the pair bonding between male and female?
Yeah, I think it's a similar thing from this like reinforcement of interactions with, in the cases of pair bonding, but here it's in the case of like interacting with your offspring. And so what we've seen, the data I showed you was that when a parent kind of interacts with a begging tadpole, that these endogenous opioid neurons become more active, and these endogenous opioid signaling systems, are released into these, different brain regions and things like that. And so we're thinking that it's this like reinforcement of like going to the tadpole and when the tadpole is like doing this begging behavior. And so this also, I think, kind of gets, and this is something we're testing now. And so I have like preliminary data with a low sample size. So take whatever I'm about to say with a grain of salt. It could be completely wrong. But what we're starting to find is that low care and high care dads have differences in opioid receptor expression in regions that are like sensitive to opioids. And so it could be maybe, I think this is like still something we need to need more data on, that low care dads, for example, are less sensitive to opioid release than high care dads because they have a higher expression of opioid receptors in these brain regions that kind of regulate motivation and reinforcement. Does that make sense?
Yes,
Yeah.
yes.
We have like preliminary data that suggests this, but we don't have like a full sample size.
So for when those of us working with non-human primates or mammals, and people are asking us, well, what do you make of this when we're describing similar patterns, behavioral patterns with, say, our monkeys? Once again, I mean, when we have to venture into the waters of making suggestions, real-life suggestions, once again, we're looking at a system where you have to just get those fathers to have an experience that feels good. Let's call it the endogenous opioid. Probably there's some kind of sensitive window when they can do this, if you really want to have those fathers become high care fathers, right? I mean, this is another system where again, am I right in saying that it's speaking in very general terms to that kind of situation?
Yeah, I think that it could be like something more general. I mean, to me, it makes sense that like these interactions in a parent have to be reinforcing in some way. And so, but a lot of this work that has been done in mammals has been mostly focused on like systems with maternal care. So the opioid allele that predicts the work that like predicts maternal and like mother-infant strength attachment, that's done in macaques. And then, we have this like opioid signaling differences that are that, for example, are different in moms that end up having postpartum depression in humans. And so, but I think it's difficult because it's very, there hasn't been a system where there hasn't been like a research organism where the opioid system has been investigated in males in the context of parenting. And so I think it's like difficult to say like, I don't think, it's difficult to say whether that's happening in mammals, but to me, it makes sense. I mean, it's happening in these contexts of maternal care, and then we see it here in our frogs. And so, something has to be reinforcing about interacting with your offspring. And so if I had to make a guess, like in your owl monkeys, for example, I would guess that there would be like differences in like opioid abundance in the plasma or something like that, or receptor distributions in the brain in dads, for example.
Thank you. I see, Sarah, you've raised your hand. You're next.
Yes, I wanted to go back to a few questions ago. When one partner seems to be kind of shirking or getting ready to bail out, what is the coordination there? And I remember I was writing about this once in tamarins, where, there are these rare reports in the field of mothers who look like they're not going to have enough alloparental support, and they'll like bite off the hands of the baby, and they'll really push it off and sometimes kill the baby. And the economist, Paul Seabright, happened to be visiting us then. And he said, well, Sarah, it sounds like what economists call a coordination game. And it's when one partner kind of pulls back to communicate and also incentivize the other partner to to invest more. So it was like a form of exploitation. And,
Yeah.
you know, it's just a hypothesis, but I liked having a word for it, coordination game.
Yeah, yeah.
And we use that later in writing about postpartum depression. And I think Ed Haig in evolutionary psychiatry has kind of proposed that might be that, though I think he uses more exploitation, but I think the economists have a good concept there.
Yeah, thank you. Yeah, it's good to have a word for it because we were, yeah, because I think, yeah, that's basically happening. So that it's like, I'm not going to compensate for you and you need to do your job, kind of as a like simplify it into a way I've explained it to my kid. And so, yeah, I think that, thank you for that. Yeah,
Well,
and I think that's what's happening.
Yeah.
Yeah, great to have a word.
Do we have any? I have more questions, tons of them. But anyone else who would like to? Can you, while we wait for somebody else to come, can you share a little bit more about the basic natural history? I mean, the life cycle of these little creatures. Like, for example, how long does it take to go through one reproductive cycle? And what kind of limitations do you have in captivity? I mean, can you go? How many generations can you go through a year, 2 years, 3 years? I mean, are you looking at things that will require several generations to examine? So just the basics of the natural history of these guys.
Yeah, so in the lab, the imitator that I talked about, it takes about 2 or 3 months for the tadpoles to complete metamorphosis. And then once that happens, then the parents can go ahead and raise another clutch. And so we usually, if we're letting the parents raise the tadpoles, so a lot of the times we just kind of like need tadpoles for experiments. And when we remove the tadpoles, they'll like lay another clutch kind of immediately. And so we can like get several clutches out of them like all the time in the lab if we're removing their babies. But if we let them take care of their offspring, you know, it takes about 2 to 3 months for them to like kind of complete this rearing. And then it takes time to get from a froglet to a reproductive adult. And this kind of depends on like how much food they have and light and things. A lot of their development is based on external factors and like nutritional resources. And so, it takes maybe like, kind of 6 months to a year to be able to complete a generational cycle. And so we're rearing a lot of the tadpoles that were high and low care now. So we're looking at like what's different about, we see these differences in behavior, but like what's different about their brains. And so there's actually quite a lot of differences in like the brain size and the different kinds of neurons and things like that. And so there are a lot of differences that on the side of the tadpole. And this is stuff that we're trying to do now, but we're also rearing some of them to adulthood to try to see if this actually impacts their own parental behavior when they become parents. So that's one thing that we're doing. Then what we're doing is some cross-fostering, because I think this comes back to the question of whether or not it's genetic or whether or not it has something to do with the way you were reared. And so, whether it's like epigenetic or whatnot. And so we're doing a lot of like cross-fostering to try to tease apart whether or not it's environment or does your genetics kind of predispose you to being a high or low care parent.
Thanks. Eugenia, you have a question.
Yes. I come from I work in the local health unit in Rome, and I work on social and on social behavior, domestic cats and dogs, and on their sexual strategies. My question is linked to the one by Eduardo, because I was impressed by the small quantity of water you showed in some of your slides. And so I thought that species at least surely lives in an unstable environment. So I was maybe I missed it, but I was thinking, how can they do all things in, you said months sometimes, but the cycle is too long for the small quantity of water. I don't know. I was just curious.
Yeah. So I think that so one, I think access to water pools that are stable is definitely like a driving force in the kinds of parental behavior and reproductive systems that they have. So species with access to like large pools of water can just like drop their tadpoles off and like they'll be fine, they'll develop, and that's where care ends, and they can do lots of clutches. And so versus like there's also a lot of predators in there. So then there's this thought, and Kyle Summers originally put this forward, it wasn't us, but that you can utilize these very small pools of water within plants as a great like hidden nursery that's protected from predators, but then there's nothing to eat, and so mom needs to be involved in some way. And so I think that but this is also like the rainforest, and so it's like raining kind of daily, and so I think that there's a rainy season and there's a dry season, and I think that actually their ability to kind of utilize different water pools kind of might dictate whether or not the species can reproduce year round, or whether or not they reproduce only in the wet season when it's like constantly raining and replenishing some of these pools. And definitely like access to pools and like kind of this like kind of niche partitioning based on the kinds of pools that they're using is definitely happening when we look at what species are where and what kinds of pools they're utilizing for their tadpoles and things like that. It's very much tied to this water pool idea. Yeah.
Thank you.
Adriana.
Hi, thank you, Laura, for such a nice talk. I am Adriana Maldonado, professor at Universidad del Rosario in Colombia, and I work on social systems in general, social structure mostly. I have a question, and you kind of already respond to it in a comment you made, but I was curious about how heritable is parental care in your system. So if you were to, create lines of high parental care versus low parental care, because you have this beautiful like variation in the behavior, could you do that and like think about like more complex cross-clustering experiments based on this?
Yeah, I think we could do that. We could create high care and low care lines if it's genetic. I think what we've seen is that sometimes it's like not very clean, like I am always a high care dad or versus always a low care dad, because sometimes when we repair them in the colony, they can sometimes, not all the time though, switch phenotypes. And I think it basically depends on who they're partnered with. So there's this kind of parental synchrony thing happening and whether or not the pair likes each other. And so there's this like kind of added complication. But I think, it might be possible. So that's one way to do it. The other way we've been doing it is kind of creating, we could create lines. We're also trying to create kind of hybrids between closely related Renitomea. This is the genus that we're working on, species that have different kinds of parental care or like begging or not begging to kind of trace the genetics. So that's more of what we've been focusing on because it's more of like a yes, no. It's like a more robust output. And I think like more easily kind of traceable genetically. And then this like variation in like who they're paired with and whether or not they're high care or low care kind of makes me nervous about like the investment that it would take to create high care and low care lines. But I think like if we looked at, I think that would really depend on the results of our cross-fostering experiments with tadpoles. Because I think if it's like we see a genetic signature, then I think that high care, low care line would be really worth doing. And I think that's a fabulous idea. But I think if it's like just based on the care that you received, like as during development, like seems to be the case in some mammals, then I think we probably wouldn't do that. So I think like these cross-fostering experiments that we're doing is actually really important in deciding whether or not we can do something like that. But yeah, it's a fantastic idea.
Cool. Very nice system. Great, thank you.
Well, I guess I go again. I have some very, very concrete questions. I like learning about how my colleagues handle their systems. So the first one is, these are very basic questions. When you do the control for the ablations, are those control both you do the ablation but do not inject? So what is the control technically in your protocol when you do the ablation of the dopamine neurons? And then the other one, very related to protocol and how we develop different systems. I got the impression that for most of the last part of the talk, when you had all the experiments, the sample sizes for each of the variables were about 8. I'm assuming those are 8 individuals, give or take a couple. But I was curious whether most of your studies are terminal studies, or if some of these manipulations allow the animals to be used again in experiments. I'm just trying to get a sense of the logistics of having a frog system in the lab.
Yeah. Okay, I'm going to tackle your dopamine question first. How do you ablate dopaminergic neurons? And so you either inject this, so there are different kind of controls you can have. You can have like a brain region control, or like, so that you're counting like the number of dopaminergic neurons in one population versus the other to make sure it's like a very specific targeting. You can also, and I didn't show you this, but inject saline instead, or whatever the vehicle was for your toxin. And so I chose to show you these like differences in brain regions to show you that like one had an effect and one didn't. But also the other controls that we've done is like looking at the amount of time it takes to be able to ablate the dopaminergic neurons, and then having this like saline there to show that when you just inject it with saline, like the dopamine neurons are fine. That's not necessarily a terminal experiment, because you could like ablate the dopaminergic neurons and then do the behavior, but to really know whether or not you killed the dopaminergic neurons, you really need to be able to like sacrifice the animal and like section the brains and things like that to really be sure. And then wait, what was the other question? Whether or not
give or take, I mean, what are the limitations? What sets the maximum number of units or of individuals that you can have in any one study? Is the per diem cost? Is it Diet Cook? Is it the size of your room? So really, yeah, I mean, what is the limiting factor here?
Yeah, I mean, people's time, and then like a power analysis on like how, like based on the variation in our population, like how much do we need to, like how many animals do we need? Because we don't want to use too many animals. I mean, we have like a responsibility to reduce the number of animals that we're using per group. And so, you know, especially if it's a terminal experiment. And so it's a little bit of like space issues and like how many couples we can have at any given time. It's about like people's time, because like, like all of you know, behavior takes a long time. But then when you're adding on like, you know, looking at the brains, counting all these neurons, you know, then like you're just like adding a bunch of like kind of other experiments at the end. I mean, that's like a year of a grad student's life. And so it's like half based on like how, like, what is the variability in our data set and how many samples do we need, but then also like how much time is somebody actually putting into like quantifying all of these like cells. And then, yeah, but like amphibians generally, I think are like, they're very easy to have in the lab. The per diem costs are like, I pay, I think like $10 per room per day or something like that, because we take care of all of our animals. And so I don't pay these like high IACUC costs because we like feed them flies.
So you don't need that. That's very interesting. So for amphibians, those working with amphibians haven't been hit yet by the fact that you have to hire all kinds of vets, and so we'll be hiring psychologists and therapists for the animals and all that. It hasn't got to amphibians yet.
No. So we do have veterinary oversight. Like if we need a vet to come look at something, they'll come. We get randomly inspected to make sure we're doing things appropriately. But no, we don't have this like major oversight, like especially for people who work in primates or even in rodents. So it's much cheaper. There's a lot less, you know, kind of paperwork and personnel.
I was talking to Steve Chang, you must know him, but it was sad. It was sad to hear. It isn't just only about economics, but he was telling me how how restrictive has become to allow students. So now you have a non-human primate colony, and it's becoming impossible to really train undergrads, definitely. And PhD students, it's becoming harder and harder. So it's not just, even if it were free, suddenly it's becoming more and more complex to make of those colonies a learning opportunity for younger colleagues.
Yeah, I agree. And I think that's why a lot of like primarily undergraduate institutions, for example, are really looking for people who do like aquatic animals like fish or frogs and things like that. Because like, as I showed in my presentation, like a lot of that work was done by undergraduates. A lot of graduates are in the lab, they do these honor thesis. It's like it's very simple, and they it's not hard to get them to like sign up to do something and to get them trained.
It's hands-on, yeah, you know what I mean? That's the experiential learning we're talking about all the time, only that then we cannot, it's becoming impossible to actually do it.
Yeah. Yeah.
Fascinating.
And I think the other question you asked me is about terminal experiments, like
No, I was wondering about you. I think you've addressed that, the extent to which some of these subjects may be part of several studies, and you did talk to that, yeah.
Yeah, I think it depends on the question. I think when we do a lot of field work, we try very hard not to sacrifice animals in the wild, like in nature, you know, and try to do that only in our lab population. So we try to develop a lot of non-invasive techniques to like measure hormones or behavior or something like that in the lab as much as possible. We've also now developed, this is a postdoc in my lab, Najwa Akbari. She has developed ways to like image tadpole brains and like neural activity in tadpole brains without sacrificing them. So we could look at activity like as they're giving different stimuli and like across development, because the tadpoles are like somewhat translucent. And so we can directly see into their brain and do calcium imaging, just like a zebrafish. And so it allows us to do this like non-terminal measurements of neural activity, which is also nice.
Guys, we're a small crowd, so please, this is the beauty of us, of Lauren volunteering her time. Don't hesitate to start your video and just jump in if you have any questions or comments. Anyone? Then I'll ask again, what have you learned about what do you know about kin recognition? I mean, we're talking about parents, we're talking about offspring. You know your parent and offspring because you're breeding them. But do we have any evidence or understanding of the fact whether there is kin recognition, or this could just be familiarity where the male would be providing care to any tadpole you bring close to him?
Yeah, so this has been work that has been primarily done by Ava Ringler. Yeah, she's done some really great work on like parental recognition of clutches and femorales. So what she has found is that it's the location that really matters, not who it is. And so there seems to be no like direct kin recognition from parent to offspring, like parental recognition, because like where you put them really matters. So males will take care of kind of any offspring that's in their territory, whereas females will like compensate when males don't provide care, but only if it's like in the exact location where they've laid the eggs. And not any else, but like who is there doesn't matter. And so this has been really nice for us because, well, one, we've showed that this, like what Ava Ringler showed in femoralis is very similar to what's happening in imitator. So they recognize it based on where they left the offspring and not who is there. So it lets us do these cross-fostering experiments with tadpoles. And it also allows us to replace the tadpole with like a robot. And then we could like turn the robot on when the parent visits or, and off and things like that. And so that's been really nice for us actually to be able to manipulate the system that parent recognition is based on space and not like direct, like who it is.
Right. That's great.
Yeah. So that's like the parent side, but then the offspring side, we've shown that, that's mostly olfaction based. And so they use olfaction when they're tadpoles and then spatial location when they're adults.
But olfaction, did they ever look at whether olfaction has a kin signature? Or is it also,
oh.
or is it both?
From in the adults,
Yeah.
with the adults. I don't think I'm not aware of any sensory system work that they've done, although I do know that they're doing a lot of this kind of work and like flexibility in parenting now. So yeah. If you haven't had her as a seminar speaker, I think she would also be loved.
Yeah, absolutely. Adriana, please jump in.
Yeah, no, I have a question regarding the spatial memory. So have you tested how good are they at remembering the spatial location? Like, do you have ones that are super good at remembering and then others are not?
Oh, yeah. Oh, wow. Yeah, that's a great idea. It would be interesting if like low care and high care were explained by differences.
Yeah, because I was wondering, like, if the reason I'm not a good parent, it's because I just can't remember where I left my kid.
Yeah, you know what? I have never really thought about that.
It's so hard to disentangle like one thing and the other, right?
A great idea. We haven't done that. I think you know, some of this work has been done by Jenny Steinowski, who's in Costa Rica. And so she, with Ophaga familio, has shown that like if you kind of move it a couple centimeters like away from where they place their tadpole, then they won't take care of it anymore. So it does seem to be like they're very attuned to the spatial location, but as to whether or not there's like individual variability, my guess would be yes, because you know, the person who's working a lot on spatial behavior in poison frogs is Sabrina Burmeister, who does these like spatial learning mazes and who's done a lot of good work. And then also Andreas Pasukonis and Bibiana Rojas in Vienna in Lithuania. And so they're starting to do a lot of these, like, how do frogs find their way and what individual variation is. And I think that there's definitely individual variability in like performance in mazes and things like that. And I love the idea that could explain, it's very interesting, but it could explain maybe some of the differences in our parenting assays.
That'd be cool.
Yeah, thank you for that idea. That's lovely.
Do we have any other questions? Lauren has a full schedule of meeting with some of us. So anybody else? If not, we could also, I mean, I'm sure she'll appreciate it. She has changed time zones big time. So we can give her a break and then she'll start with her meetings at 1. Anybody else with any questions? If not, I think we can thank Lauren for a wonderful presentation and session of questions. Thanks to our YouTube community. I don't know how many of you are out there, but remember that we also always welcome you accessing the Zoom meeting. There are no passcodes. It's open to anyone, so please don't hesitate to join us if you'd like to be with the Zoom community. So with that,